Hello dear students, today we will take up part 1 of chapter 1 of class 12th biology, sexual reproduction in flowering plants. The learning objectives of today's session are to understand the concept of sexual reproduction in plants, to know about the structure of microsporangium and megasporangium, to understand the process of microsporogenesis and megasporogenesis, to understand the structure of pollen grain and its economic importance, to understand the structure of flower and various pre-reproductive events which leads to formation of pollen grain and embryo sac. Students, you must have query that why sexual reproduction is so important. So, let us find answer of it. You have studied in class 11th that sexual reproduction brings variations and increases survival rate of any organism. That is the reason why all the higher organisms resort to sexual reproduction. As you reach your puberty, you know that hormonal changes bring changes in our body structure. Same with plants also. Hormonal and structural changes in plants leads to development of a flower. You must have studied structure of flower in your earlier classes. Now, we will talk about reproductive parts of a flower. Androecium. It consists of whorl of stamens and it represents the male reproductive organ. Gynoecium represents female reproductive organ. In this diagram, we can see parts of a flower. Sepals and petals are accessory parts. Stigma, style and ovary forms the gynoecium and stamen forms the androecium. Both are directly involved in sexual reproduction. If you observe a typical stamen, it consists of two parts, long and slender stalk called filament and terminal bilobed structure called anther. It is very easy to observe it in our surroundings. Just analyze flowers in your surroundings and I assure you that you will surely enjoy the variety of flowers. Anther is bilobed, which means it has two lobes. Each lobe has two theca, that is why it is also called dithecous. Each anther contains 4 microsporangia. When microsporangia matures, it becomes pollen sacs and are packed with pollen grains. We can see two diagrams. Diagram A shows stamen and diagram B is a sectional view of an anther in which we can see 2 theca and 4 microsporangia. What is line of dehiscence? It is the part of the anther which dries up and cracks, thus releasing pollen to a long distance. Let us discuss what is inside the microsporangium. Each microsporangium is surrounded by 4 wall layers: epidermis, endothecium, middle layer and tapetum. The outer 3 layers are for protection. Endothecium also helps in dispersal or dehiscence of pollen grain. Very important function is performed by tapetum, which is the innermost layer. It is multinucleated with dense cytoplasm. It nourishes the developing pollen grain. If it gets malnourished, then we will not get viable pollen grain and sexual reproduction will fail. The center of each microsporangium contains homogeneous cells called sporogenous tissue and it contains spore mother cells which are diploid in nature. In this diagram, you see diagram A which shows young anther, diagram B which shows all the 4 layers properly and diagram C shows the process of dehiscence. Students, let's talk about the process of microsporogenesis. It is a very important process. The sporogenous tissue of microsporangium differentiates into diploid microspore mother cell or pollen mother cell. These cells undergo meiosis and form haploid microspore tetrad. Haploid means the number of chromosomes will be half in daughter cells. As the microspore dehydrates, it dissociates and forms 4 microspores. Each microspore develops into a pollen grain. This process is called microsporogenesis. Now we will discuss about the structure of pollen grain. Pollen grain represents the male gametophyte. It is spherical and measures about 25 to 50 micrometer in diameter. It is covered by 2 layers. The hard outer layer called the exine is made up of sporopollenin. It is the chemical which can withstand high temperature, strong acids and alkali. No enzyme can degrade sporopollenin is so far known, and that is why pollen grains are also obtained as fossils. Now question arises, if it is so hard, then how it will release its male gamete for the process of fertilization? The answer is germ pore. The exine has prominent apertures called germ pore, where sporopollenin is absent. Germ pore helps in development of pollen tube. The inner wall of pollen grain is called entine. It is thin and continuous layer made of cellulose and pectin. Students, see the diagram showing the structure of pollen grain. We can see outer covering exine. On maturity, the pollen grain contains 2 cells, the vegetative cell and the generative cell. The vegetative cell is bigger, has abundant food reserve and a large irregularly shaped nucleus. This cell helps in formation of pollen tube for the process of fertilization. That is why it is also called tube cell. The generative cell is small, floats in the cytoplasm of vegetative cell. In 60% of angiosperms, pollen grains are shed at this 2-celled stage. In others, the generative cell divides mitotically to form 2 male gametes before pollen grains are shed. Thus, a 3-celled stage is formed. We can see the structure and development of vegetative cell and generative cell in this diagram. Diagram A shows pollen tetrad. In diagram B, we can see the changes occurring in the pollen cell. It increases in size and nucleus becomes prominent. In third diagram, nucleus becomes asymmetric, spindle shaped, and in last diagram, mitotic division occurs which forms vegetative and generative cell. Now, we will talk about the economic importance of pollen grain. You must have observed that during the season of spring, many people complain about sneezing or coughing, because at this time many pollen grains are in the atmosphere and pollens may cause severe allergies and bronchial afflictions. It may cause chronic respiratory disorders like asthma, bronchitis, etc. Pollen grain of Parthenium or carrot grass causes pollen allergy. Students, this was the harmful aspect, but pollens could be beneficial also. Pollen grains are rich in nutrients, hence used as pollen tablets for food supplements. Pollen consumption increases performance of athletes and race horses. Some other features are, after shedding, the viability of pollen depends on temperature and humidity. Viability means capability to bring about fertilization. In wheat and rice, the pollen grain lose viability within 30 minutes of their release. In Rosaceae, Leguminosae and Solanaceae, they remain viable for months. Pollen grains can be preserved for years in liquid nitrogen. It can be stored in pollen banks and used for cross-fertilization for better varieties. In this slide, we can see various products formed by pollen grains like tablets, syrups, etc. So, students, this is all about the male reproductive structure and formation of pollen grain. Now, let us talk about the female reproductive structure. The gynoecium represents the female reproductive part of the flower. The unit of the gynoecium is carpel or pistil. It may have single pistil or carpel, monocarpellary, or may have many carpels, thus called multicarpellary. The pistil, if more than one in number, may be fused, and such pistil is called syncarpous, as in diagram B. If pistils are free, then the term used is apocarpous, as in diagram C. Now, let us talk about the pistil. Each pistil has three parts: the stigma, style and ovary. Inside the ovary, there is an ovarian cavity which is also called locule. It has ovules or megasporangia arising from the placenta. Ovule is also known as megasporangium. The number of ovules inside the ovary may be single or many. Students, let us learn more about ovules. Ovule is a small structure attached to the placenta by a stalk called funicle. The body of the ovule is fused with the funicle in the region called hilum. Hilum is the junction between the funicle and ovule. Each ovule has one or two protective envelopes called integuments. Integuments covers the ovule except at an opening at the top called micropyle. Opposite of the micropylar end is the chalaza, representing the basal part of the ovule. Inside the integuments, there is a mass of tissue called as nucellus. The female gametophyte or embryo sac is located inside the nucellus. Students, let us learn about the process of megasporogenesis with the help of this diagram. In the first diagram, we can see that one of the nucellus cells towards micropylar end differentiates into megaspore mother cell. MMC is a diploid cell having dense cytoplasm and prominent nucleus. The MMC undergoes first meiotic division, resulting in formation of two haploid megaspores, which is called dyad. And after second meiotic division, 4 haploid cells are formed, which is called tetrad, and they are arranged linearly as we can see in third diagram. Out of the 4 megaspores, 1 megaspore is functional and the other 3 degenerates. One functional megaspore develops into the female gametophyte. Female gametophyte is also known as the embryo sac. Now, let us talk about the development of embryo sac. The nucleus of the functional megaspore divides by mitotic division to form 2 nuclei, which move to the opposite pole, forming 2 nucleated embryo sac, which we can see in fourth diagram. 2 successive mitotic divisions leads to formation of 4 nucleate and later 8 nucleated stage of the embryo sac, which we can observe in fifth and sixth diagram. All mitotic divisions are free nuclear type, that is, karyokinesis is not followed by cytokinesis. It means only nucleus divides but cell does not. But as the embryo sac grows, 6 of the 8 nuclei get surrounded by cell walls and organized into cells. Let's see their rearrangements. 3 cells grouped together at the micropylar end constitute the egg apparatus. Egg apparatus consists of 2 synergids and 1 egg cell. Synergids have special filiform apparatus which play an important role in guiding the entry of pollen tube into the synergids. 3 cells arranged towards callosal end are called antipodal cells. The large central cell has 2 polar nuclei. A typical angiosperm embryo sac at maturity is 8 nucleated and 7 celled. This is the write-up of whatever we have discussed just now. Dear students, now let us have a quick recap. We have understood the term microsporangium and megasporangium. We have learned about the process of microsporogenesis and megasporogenesis. We have also learned about pollen grain and embryo sac. We have understood the structure and importance of male gametophyte and female gametophyte. Dear students, note down your assignment from today's session. Where is sporopollenin present in plants? State its significance with reference to its chemical nature. An anther with malfunctioning tapetum often fails to produce viable male gametophytes. Give reason for this. Describe the development of male gametophyte in angiosperm with the help of a suitable diagram. Explain microsporogenesis in flowering plants. What do you understand by egg apparatus? And the last question, describe the structure of the embryo sac of a mature angiosperm. Explain the role of synergids in it. With this, we have now come to the end of today's session. I hope you have understood the topics and also enjoyed it.